different places, and if there was a little bit more money invested, we could have seen, instead of testing 20 or 30, and since there are so many countries that come into that hab, you could find fungus from all over the world. Wow. Okay, so my project was fungal contamination in the Mars Desert Research Station, and I'm Alice Yount of crew 139A. My school, McLennan Community College, when we go, we have 2 crews, so instead of spending 2 weeks there, we have 2 crews who spend 1 week there. And I got the first half. So there was definitely some stuff I had to do before I got there. First of all, there's paperwork. There's always paperwork. So much paperwork, but it was worth it. And then I had to research and plan my experiment. First I was going to be doing bacteria, and then I decided, you know what? It was brought up to me that I could do something with fungus, and I've never studied that before, and I was like, you know what? That's an opportunity to learn something new. So I researched into that and learned some really interesting stuff. And because it's through our school, it's actually a class. It's undergraduate research. So we had to be up at the school every Tuesday from 6 to 9.
In the morning or
At
at night?
night. And yeah, it's definitely PM.
Oh, that's good.
And something special about our specific group was we got our ham radio license, which means that when we were out on the walkie-talkies, we could talk to each other from longer ranges, which was definitely helpful when we went on our EVAs. And that took 2 4-hour sessions. I had to take a test a couple times. And I finally got my call sign KG5AWG. And that was the most rewarding thing I felt in a while. It took so much work. And then finally, I was in charge of the biology EVA, so I had to prepare like all the ingredients I needed to teach everybody how to gram stain. And then because I was going to be using fungus, technically it's biohazard, so I had to fight FedEx on how I was going to ship them home, because I obviously couldn't take them on the plane. They would have thrown a fit, which FedEx threw a fit, but we eventually figured it out. So what is MDRS? It is the Mars Desert Research Station, and it's preparing for the great next adventure, which meaning we want to go to Mars, and we don't want to just walk on the moon. We want to actually stay there for 6 months at a time, a year, 2 years, colonize it. So before you do this, you have to overcome some things with space travel, like survival, food, water, air, and psychological issues that are going to arise. Because when you're with the same people, around 6 people in a tin can for 6 months, you're going to go crazy. And you got to deal with these people, and you can't just open the airlock and push them out. You have to actually learn how to exist. So this is a picture of the tin can, as I call it. That's the habitation. And a little bit further, that's the green hab, where we actually grew things. So the one we went to, there's several all over the globe. The one we went to is in Utah, and it's cylindrical with 2 decks, which are 8 meters each. It's built to house 6 people, and it's research for exploring Mars. And what a lot of people don't understand is you don't have to be just a science major. There's biology, chemistry, architecture, human studies, food studies. And on my crew alone, we got the fungus studies. We got the human studies, somebody with a quadricopter studies, and the effect of food on digestion. And this is actually a map of everything. So that was the downstairs, and you can kind of see how everything would look. It's cylindrical and, oh, it's kind of cozy.
It's actually round, which is cylindrical, but the picture is kind of oval because of the frame-rated computer, not because of proofreading.
Yes. So there were certain things that I had to take care of while I was on Mars. First of all, we had to clean up after ourselves. We're not heathens. There was cooking we did, which was surprisingly really fun. We had fresh bread every single day. It was amazing. And I'm pretty sure that I gained weight there. Most people were like, it's going to be disgusting. It was good. Bacon, Spam, Velveeta cheese. All I had to be was shelf stable. And then, of course, I was testing for my project. And then I taught my lab, which was the biology EVA. And then every single day, or at least 3 times a week, we were supposed to blog. I did it every day. Let's see. And then we did our EVAs. We went to the dinosaur quarry, which we actually found dinosaur bone chips. So cool. And apparently you figure out that it's a dinosaur chip by sticking it to your tongue and seeing if it sticks. I did it a couple times. And then there's Candor Chasma, which is basically a huge canyon, I guess you could say. And within it, there was sheets of gypsum like the size of plates. And we got to actually chip it off the walls. And then we did the chemistry EVA and the geology EVA while we were in Candor Chasma. And that's me and Michael while we were at the dinosaur quarry. Okay. And we actually took this picture when we went on a geocache, which was basically somebody left a pile of goodies from another crew and we were supposed to find it. And this is on top of the mountain where they hid the clue. And you can see that you can't even see the hab. We walked all the way in there in our spacesuits, walked all the way up, but it was terrible what they did to us. I'm not even going to give a hint in case somebody else goes and finds it. It's terrible. Okay, so part of my project. Why I picked it was I actually learned that fungus are everywhere. They're almost as prevalent as bacteria. What's so special about fungus? They can destroy property, they can cause asthma, they can cause skin problems. And what becomes really deadly is when somebody with a suppressed immune system gets a hold of fungus, it can turn really, really bad. And there's been studies that have suggested that space travel actually suppresses your immune system. Now, when you have close quarters, multiple people living in one place and you're not going to be getting out very often, you have the possibility of fungal growth, and with a suppressed immunity system, it could get bad really fast. James?
Maybe you pointed at the computer instead of the
Oh,
screen.
my bad.
I don't know if that works or not.
It's okay, I'm just a little special. So something about fungus that I learned was that there's two types. There's yeast and mold. And mold is what you're usually going to think of when you think of a fungus, like something growing on a fruit or on the wall. It's aerial because of these hyphae, and the little dots are actually the sexual parts which create the spores. And those are just yeast. They're just going to be little single-celled, so they're not going to actually have the height that you would think a fungus would have. So here's my project. Okay. So I had high-tech contact slides with these specific ingredients in them. They were to suppress the bacteria that was found in the place and only let the fungus grow. And I tested using the manufacturer's protocol. And I sampled 6 sites, 3 upstairs and 3 downstairs. And I sampled on the first day we got there before cleaning. Then I sampled day 7 before we cleaned, and then after we cleaned. Then after I tested them, I put the high-tech slides in their plastic container, and I put them at room temperature until the 8th day they were shipped. And then upon arrival, I photographed my high-tech, and then I recorded the fungal growth. And the certain colonies that I wanted to keep, I plated on potatoidextrous agar, which is basically food for fungus, to further study them. And these were 2 I found. Figure 7 was a fungal colony I subcultured from the site 1 at the kitchen counter. And figure 8 is what it looks like under the microscope. I used a lactophenolcotton blue staining to look at it, and you can see here these branches, and there's little phyllodes, and that's typical of the Penicillium species. Okay, so site 1. On day 1, we found very little. How I did mine was 0 contamination was obviously 0 fungal colony. 1 would be low, which would be 1 to 2. 2 would be 3 to 5, and 3 would be high, which was 6 and above. So as you can see on day 7, since this was the kitchen, we kept it pretty clean because that's where we prepare our food, which didn't surprise me. Site 2, there was contamination day 1 and day 7, but apparently the cleaning did pretty well because it lowered in severity. Site 3, you can see that the fungal contamination slowly decreased, and site 1 through 3 is my upstairs. Site 4, the contamination stayed regular all throughout. Site 5, it decreased, but since it was so high, went from severity to moderate. And then Site 6, it was contamination, the same throughout the week. My hypothesis is that since Sites 4 through 6 were downstairs, they were readily being contacted from the outside, and that's where we had our shoes. So most likely that was the most shuffling around. So in conclusion, when we arrived at the habitation, it could have been cleaner, I'll admit that. And I actually was worried that I wasn't going to find anything, but when we were doing some further studies, looking around, we actually found 2 spots that had already growth. These were the most common colony types we found: Penicillium, which is penicillin, Zygomyces, which is a common bread mold, which wasn't uncommon since we were making bread every day, and then an unidentified acceptated fungus, which isn't uncommon because the way that you do your testing, you tease mat it, which means you have your fungus on your plate and you take it off and you put it under a slide. And you put a lactophenol cotton blue stain, which is something to look over it, and you put it under a microscope. And when you look at it, you can only see certain characteristics. And sometimes, like a septated fungus, it could have 5 or 6 different things it could be. So then you have to go back to the colony morphology, which is how it looked, and you kind of have to play around with that to see if you can get the right thing. And that process takes hours of hunched over a microscope. So the largest amount of fungus we found was downstairs, figures 4 through 6, which, as I said, it was most readily accessed from outside, which didn't surprise me. And the traditional means of identifying them, I used the tease map and the lactophenol cotton blue, which is traditional. That's what you usually use. And then future opportunities, I took all the fungus I had, and the ones I chose to subculture out, I did 10 plates. And then in the future, I did DNA testing. So here's some post-Mars responsibilities. A part of the class, I had to present my results to the Scholars Day at our community college, and as well as to my advisor's class. She was in the MLT program. And then I had the possibility of going through DNA sequencing to further my research, which I got very lucky that the funds were available, and I used a Carolina kit to actually sequence what I found. So first of all, you have the Carolina kit, and there's 3 steps. You have DNA extraction, and you use the ITS2, which is the internal transcribed spacer 2, which basically means in all this information, there's a certain part in the cell that has the DNA. And I use the ITS2 for fungus to pinpoint on where the DNA would be. And then when it was extracted, then I went through the process of amplifying it through a polymer chain reaction, which is a PCR product. Then from this PCR product, I did electrophoresis with gel green stain. And what this is I'm actually going to go ahead a little bit and show you all. That's the electrophoresis. I made my own gel. I had to make it a certain thickness. Then I took pipettes and I took my PCR product and I put it at each of the little slots I made in the gel. The first slot was used as a DNA ladder, which a DNA ladder is saying this is what DNA should look like. When you go through this process and put it under a special light, you should know that the DNA ladder has something. And sometimes if you make the gel too thick, if you can't see the DNA ladder, you know you screwed up and you have to do it again. So you pour certain liquid over the gel and in this machine, and it sends electricity through it, and it makes the PCR product and the DNA go down the gel so you can look at it under a light later. So once I was able to look at the gel under the light and I could see that there was PCR product it was the stain made of orange under the UV light, I sent all my work to GeneWiz, and they put it through their machines and they were able to decode the A's, the G's, the U's, the T's. And from there, they gave me this whole paragraph of all these A's, these G's, these T's, and I was supposed to put it through the BLAST and the NCBI to figure out what it was. So I put 10 through the process, and because of some issues through the mail, I got 4 definite ones and 6 that were unsure. There wasn't enough information to code it correctly. So I got Epicorm nigurium, and then this one I got from site 5. And what this is a common plant fungus. Then I got Aspergillus species, which was from site 6. I got an uncultured penicillium clone. And then the most interesting part was, as you can see, AY10. That's the one where I got from the wall that was already growing, and I learned that that's Penicillium cordubitans, and it had visible fungus growth. All the rest of my species that I picked up was in spore forms, which mean they were microscopic from the eye. I was testing sites to see if it was there. The Penicillium, the last one, was stuff that was already shown there, which, because I was the health and safety officer, kind of concerned me. We're in a small space, we're living there for a week, we're breathing this in every time we go to these certain spots. It shouldn't be here.
Sorry, what wall was that? Yeah.
It was, I can actually show you. I'm just gonna take, sorry. I can show you exactly. Because, oh, there it is. All right, so you see the EVA prep room, which is right by the airlock.
Yep.
This is where you come in the entrance. It was there was suits that were hanging right here, and it was on this wall. And it was also right here. Yes, so they told me to get some bleach, and I cleaned it up.
Well, that was one thing I was going to ask questions about.
Want me to do it?
Yeah, if you could just, sorry. Yes, there you go.
So you test the room, I guess that thing, what about the soup parts? In other words, notoriously they're, they get sweaty and they keep moisture and they're being cured and...
Well, I really can't blame MDRS for this, although the building is old and it probably should be cleaned a little bit more readily than it was, because just the fact it's old. The way that fungus are, they're opportunistic, which means if you give me all the right food and the right environment, I'm going to grow. Inside, where the contamination was, it was places where there was a drippage from the sink, or it was a drippage from the AC unit. There was obviously, the walls were damp, and since you were inside, it had a moist and a warm environment that really, it could grow however it wanted.
So then should the green have be looked at too?
Well, the green hab is, I think there are other issues that should be addressed for the green hab other than fungus, but that's besides the point. I mean, don't get me wrong, I had an amazing time at MDRS. It was full of these experiences, and I shared it with 6 people that I didn't know before, and I think of as good friends today. Being the biology officer and the health and safety officer and learning so much about fungus, it was very enlightening. And when we actually did the biology lab, we couldn't look at any of our samples to barcode it, no, not barcode it, but to gram stain it, because it had fungal contamination on every single plate. It was like, well, here's how you do the gram staining, even though we can't do it with the stuff we grew. Now let's go burn it. You have to autoclave it, and we were told to do it in the oven, but the team after of us decided to burn it outside, which was a much, much better decision.
You can't do that on Mars.
You'd find another place other than the oven.
Oh, yeah.
Michael can attest to this. It was terrible smelling. When we burned all the little bacteria.
Oh,
Yeah, Brad could remember too. These are my references. Do you have any questions? Sure.
Why did you choose not to do a sample after cleaning them the first time?
Because I had a limited amount of the high-check media, and I decided that it was better to try on the last day, because we had been living there the entire week, and the results was more of us living there than of the previous crew.
Okay.
Did you have a question?
Did you run into any scorpions over there?
No, we did not run into any scorpions, but we did find Nutella was excellent rat bait. Yes, sir.
How much of this experience, you think will actually translate to a real Mars have on Mars?
Well, I think it's a definite translator because even NASA, they have different machinery. They actually much advanced. What they do is they swab an area and they put it in a machine and it tests for gram-positive, gram-negative bacteria and fungal stuff. And the fact that the astronauts are worried about it is definite. That is something we're going to have to worry about because you can't really get rid of all the fungal spores. There's going to be stuff with you, and when you go and you live in a habitation, you're going to live there. And no matter how clean you keep it, there's going to be growth, and you need to learn how to, or not learn, but you need to be aware of the situation enough to, I need to clean up this often. I at least need to test this. Because what if you get there and your immune system is compromised? Well, if you're staying there for years, you can't exactly go back. And these fungal spores, there is certain ones that can cause tuberculosis in those who have suppressed immune systems. And how much are you going to be bringing to Mars when it comes to medical equipment? That could get really dangerous really quickly. Yes, April?
Do you have any maintenance recommendations based on your work?
I do. I would recommend that you at least search the walls, especially the ones that have continued drippage, for fungal contamination. And it's actually recommended by others. I was looking online when I was told to clean this up, that you need to check it every so often to make sure it hasn't grown back. The nature of fungus is they have so many little spores. When I was working with it under a HAB, under safety level 2 biohazard lab, I was under the, with the air vents and everything, under a slide. Sorry, I'm not describing this very well. But I would take it open to test it and to put it on another subculture to study it later. Well, spores you can't see, but they get everywhere. There was one set that I almost had to throw out because a ton of them were contaminated. And it was really frustrating because I sat there and I bleached the area really good before I went on to each sample. And it's just really easy for these little spores to get everywhere and you don't have to see them until they're fully grown. And by that time, you're already breathing them in. And if your immune system is compromised, then you get back to this whole cycle. Did I answer your question? Any other? Yes, sir.
Two. First, I was wondering about that. There's an attic area also up there. Was that included, or that should be looked at maybe? And second question.
Actually, I would agree with you. That area should be looked at because of the fact that the water tank is there. So you have constant source of water that could maybe leak and could possibly be a place of fungal growth. Although I did not look there because of the fact I was trying to concentrate on 3 areas top, 3 areas on bottom, and honestly, I didn't go up into the attic place with the water tanks.
So something for the future, especially if it's a place that people don't look at.
Exactly. It could be growing there unsuspected.
Second question is, we answered about maintenance recommendations. What about have things that they should do, not to the design, but I mean modification? Do you have any recommendations for any modification that they should make to this? Because this is all.
Yes, I think.
Safety's number one.
I understand that the MDRS program isn't working with a lot of money, but the fact is, there was rats inside the HAB too. It wasn't just the green HAB. There are spots where rodents are coming in because it's the only place for miles around. It took us 30 minutes from the main road to get where we are. So obviously, if somebody's living somewhere and it's free food, why aren't rats and other creatures going to come out there? I just was really glad it wasn't a snake.
Those will be next. If they're rats.
Exactly.
snakes
You
will
either get
come.
snakes or rats.
Mm-hmm.
I don't know, it was kind of satisfying in the middle of the night here. Snap!
So you're saying
Got another one.
So you're saying they should make modifications to the HAB to.
I do think that there is definitely some patchwork, and I think that they should get somebody to go in there and maybe even inspect the building, because I wonder if it would even pass inspection. There was 3 spots in the walls that had fungal growth that was a good block. And I'm not meaning to put MDRS down. I just think that they should be aware of this if they're going to send somebody else in here. Yes, sir.
How many, we've had experience on the space station, even the Skylab, and of course, especially the Russian Mir space station apparently had problems with fungus. How much did any of this fungus kind of compare with what they've seen on those various places?
Well, unfortunately, when my samples were in the mail, they broke open and they had to be further diluted. That happened twice, unfortunately, which was very, very frustrating. And I only had enough material to do 10 samples. I think that if more funds were available to me at the time, we could have done more testing, sent in more samples, and gotten a better range of what's actually there.
But I was at a comment, the school a block from my house that my kids, in the middle school, has now been condemned by the city of Middleton because it's full of fungus. It's